Hi everyone, welcome to ISME Conversations. My name is Rochelle Sue and I'm a member of the ISME Early Career Scientists Committee. It's a great pleasure to have Professor Phil Hugenholtz today for this interview. Phil is the Vice President for ISME and the Director of the Australian Centre for Ecogenomics. He's a professor at the University of Queensland in Brisbane, Australia, and he's won multiple awards and fellowships, including a Young Investigators Award from ISME in 2006. He was also elected in 2012 as a Fellow of the American Academy of Microbiology and elected a Fellow of the Australian Academy of Science in 2017. So welcome, Phil.
Thanks, Rochelle. Thanks for having me.
You've had a really successful career in microbial ecology. How did you get to where you are now, and what is your research, and how did you decide on what you wanted to study?
Well, I think like many people, it's a combination of luck and willing to take a risk at the time, and then the rest of it backfilling it with hard work. So I think that a couple of instances, I was just in the right place at the right time, and I grabbed the opportunity. So that's the one thing I would tell early career researchers is to take the opportunities as they come up, because generally they work out well. My research, I describe myself as a microbial diversicist that dabbles in microbial ecology and dabbles in microbial evolution. So I got interested in microbial diversity during my PhD. I was actually looking at the biofilms that were growing on air conditioning systems here at the University of Queensland, in the Dewey Library, in fact. And I was working with my PhD advisor at the time, Professor John First, and I was using culture-dependent methods and getting isolates and looking at them. And I remember I really did enjoy growing microorganisms and seeing them and isolating them and seeing them under the microscope. And I started, and this was a fair while ago, I have to say. This was the early 90s, late 80s, early 90s, and molecular techniques were just starting to come in, and I was really not having a great deal of luck characterizing these organisms phenotypically. I was doing all the usual things, oxidase and catalase tests. And then John First, to his credit, he was aware of the 16S RNA, and we just started to make movements into that. And I was quite liking that because it was like solving puzzles, and I really gravitated to it. And I liked the idea that it gave you a more definitive answer. And then this is one of these lucky breaks or serendipities that happened, is that Erko Stackerbrandt came and headed up the microbiology department, came to Australia from Germany, and Erko Stackerbrandt had been working with Carl Woese, and so he was really one of the few aficionados of 16S RNA analysis, and he came to University of Queensland at that time, and so I had a really good opportunity to learn how to do that from more experienced people. And I do mention that John First was already thinking in that line, so I want to make sure I credit him with that. And I made a nuisance of myself with Erko Stackerbrandt to try and learn. I was more interested in the analysis side rather than the generating side, and he got cranky with me a couple of times as I was always asking questions. But that actually, then I found something that I was actually pretty good at, because I am doing the sequence alignment and inferring trees and getting a handle on that. And that's how I got into it. And it was through that process that then Erko wrote me a reference letter that got me into Norm Pace's group, and Norm was the guy that was the first to apply the 16S to environmental samples, and that's how I got my foot in the door. Yeah, and since then, I think probably I've already answered that question too long, so I'll let you ask another question.
Yeah. So how did you know when you wanted to start your own group, and what advice would you give EMCRs that would like to start a new lab?
I didn't really want to start my own group. I liked being a postdoc, and in fact, I stretched that on for as long as I possibly could. I did 2 postdocs sort of back to back, and then I was sort of a junior group leader, and I was sort of a group leader of one myself. And then again, by another bit of luck, Jill Banfield was doing sabbatical in Australia. She's originally Australian, and we got talking, and she invited me to come and work with her back in the US, and I took that opportunity. So again, I postponed running my own group for a while, so I was a bit like back to being a postdoc in Jill's group, and that was a great experience. So really, I didn't really start my group until I went to the Joint Genome Institute in 2000 and was it 6? I think 2006, and by then I was already fairly advanced in age, I guess. And that was my first experience heading up a group myself. And at the Joint Genome Institute, they don't have PhD students, so I began with postdocs, and I got my first 3 postdocs who were already very competent and independent researchers, so that was a great joy. And my first 3 postdocs were Victor Koonin and Falk Warneke and Hector Garcia Martin, all with different skills. So Victor was a bioinformatician, Falk was a classic microbial ecologist, had been trained in Germany, and then I guess Hector was the wild card. He was a physicist, and so he was interested in using ecology to advance his to do his ecological theory. He wanted to do flux balance analysis of communities. So anyway, with those 3, with the great, it was a bit of a cushy start to running your own group, let's say, as opposed to being thrown in the deep end and then taking a PhD student on from scratch, which I only really did when I came back from the US in 2010, and that was when I was really primary supervisor of my first PhD students. So by 2010, I was already in my early 40s. So that's the first time that I actually had PhD students, and I completely bubbled it up to begin with because I set 2 ambitious projects, and I realized that you have to start, particularly honors students, for instance, and PhD students, on simpler projects, but I've learned that since then. And it's been a great joy ever since, once I got the hang of it. So I don't know if I'm a good example, except to say that to ECRs, I would say if you have an opportunity to travel and go to different labs, that's really good for career-wise, because if you come back to your home country afterwards, for instance, like I did, that gives you a bit of an advantage, I would say.
And you've also got a startup company. What's been the challenges of establishing the company, and how do you find balancing working at the company and working as an academic?
Yeah, that's a great question. So I got into the startup company space because our president of the university, the vice chancellor, was encouraging researchers to try and translate their research. And so there was an opportunity. So together with Gene Tyson, we saw this opportunity to take some of the culture-independent molecular analysis we'd been doing, the metagenomics, and commercialize it, because the reason it was possible was because the price of sequencing had been coming down and down and down. So suddenly it became apparent, I think it became apparent to a lot of people, that it would be possible to do metagenomic profiling of gut samples, human gut samples, at a cost that you could offer it as a commercial offering. And so to be honest, in the early days, it was because I had no commercial background, I found it quite confusing, and I didn't really know what I was doing. And so we sort of bumbled through it a bit. You get some help from most universities will have some commercial guidance, and we were getting a little bit of that. But bottom line is that it really only took off when we had some business people got involved. And that would be my first bit of advice is, unless you're very entrepreneurial and have prior experience, get some business collaborators. And so you bring the science, they bring the business, and you learn to talk each other's language a little bit, and then it worked. And that's been a great experience, a great joy, and particularly the incubator phase when you've got just a few people and everybody's really excited about it. And that company's now grown to over 50 people, I think maybe even over 60 people. And so nowadays I am spending 20% of my time sort of helping with the scientific direction of the company. The company is called Microbial. It's not a commercial plug or anything. So I enjoy hearing about the science. Too big an entity now for me to stay across everything that's going on, but I'm enjoying where the science is going, and I contribute as the best I can there. And it's good to be ensconced in that. And I go out to the headquarters there and spend the time with people, and I really enjoy that. And that's how I balance it. So I do 20% during my work week and 80% for my day job as a professor and do the research. And there are different things going on, so I find it all very enjoyable. The reason that most of us are drawn to this is for curiosity and the joy of discovery, and so I get to do that on the commercial front and on the academic front, and that's a good balance. And try as much as possible to not let that spill over into weekends too much. So yeah.
Great. Was there any early challenges when you were in EMCR, and how do you think things have changed for EMCRs today?
I think it's harder today. I hate to say it. I mean, I think it's harder to be an EMCR today than when it's just there's more people, more competition, and less resources. So when I was younger as an early career researcher, I think it'd be safe to say there were more opportunities, although technologically speaking, there are greater opportunities today than when I was an ECR. And so I wasn't actually planning out my career much beyond what I was doing at the time. So if I was in the postdoc, I was focusing on doing the postdoc work, and then just things would turn up, and I've been lucky in that regard. And I'd work hard in between, and again, coming back to taking the opportunities. But I am very concerned about our ECRs, and maybe I should only say from an Australian context, because I think Australia doesn't invest enough resources into its basic research. It puts too much money into nuclear submarines, for instance. So I would like to see more money going into supporting ECRs. There's kind of this period between, you can get some fellowships early in the career, and then you can get some when you're established, but there's this intermediate ground which is a little bit tough to travel. And I do worry about a lot of talented people like for the centre here when they're on soft money, and how they can progress in a fashion without having to really put themselves at a disadvantage. But yeah, it's a worry.
What do you love most about your job?
Well, you know what I love most about my job. What I love most about my job is brainstorming and doing experiment, and it's a luxury to being the group leader in that you can get a few of those things going at once, and then you get to see how they're progressing through the hard work of your talented researchers. And I just love that discovery element to it, and I really do enjoy writing and putting papers together, and that really is I mean, I would have said earlier on in my career it was about getting the big runs on the board. That's exciting, but these days the joy is in the journey of creating the research, and I really do enjoy that mentoring element a lot. And I like to see the younger team members succeed, and I remember the thrill of my first paper, and I love to be seeing that thrill in young researchers, because then I get to sort of live vicariously through that same experience again.
Yeah, it sounds like you've got a lot going on. How do you normally take time out?
Well, actually, what I do is don't let the group get to my individual group. I don't like to let that go above, say, 10 people, 10 max. Obviously, you run a center of many more people, but then there's a whole bunch of other group leaders here, and they have their own groups. But my personal group, I like to keep to under 10 so that I can see everybody and work with everybody in a more meaningful way. I get worried about when the group, and I did go through a phase where the group, you had opportunities and you let the group grow very large, and then I felt way too spread too thin and not engaging enough, and that's really not as enjoyable. So what I do now, I think I have a much better work-life balance than I did when I was younger, and I encourage everybody here at the centre also to try and keep a regular work hours and take their weekends, particularly if they have families and young families. And what I do now is for entertainment is I go kayaking, that's fun, paddling on the Brisbane River, and I am in a music group, and so I do some things outside of science, but I do actually read papers and stuff in my spare time for enjoyment. So I do count my job as one of my hobbies. And the reason I now have these hobbies is because I was asked by an ECR a few years ago, what do I do outside of science? And I didn't really have a good answer at that point. I thought, oh, wow, I really do need to have something outside of the science. So now I do. So those are the main hobbies I have.
Good. So yeah, I guess that's pretty much all the questions. I don't know, is there anything else that you'd like to tell the ISME EMCRs before we finish up? Give them any advice?
I think the main thing I've already said is take the opportunities when they arise. One thing I would say is, and I didn't do this and I got lucky, do a little bit of research on the lab that you're going to. First of all, check that the group you're going to is productive. That's a good sign. But also, if you get a chance, talk to people within the group that are already on the ground to find out what the lab culture is like and whether it's going to be enjoyable. I think that really is important. And then when you start your own group, definitely pick your initial people carefully. That people that you're going to be spending a lot of time with them. So pick people that you're going to be compatible with. I guess you'll naturally do that anyway. Yeah, I think that's probably the best advice I can give at this point.
Great, thanks. Yeah, we really appreciate you sharing your journey with us and the advice that you give to the EMCR community. Hopefully we'll be able to see everyone at the ISME conference next year.
Yes, listen, let's hope it's all on. 22. Look forward to it.
Thanks, Phil.
Thanks, Rochelle.